to the first seminar, ABI seminar for the year. Vinod's still looking after these this year, but he can't be at this one, so he asked me to introduce Kevin, who is a Kiwi who initially did his PhD with John Butcher in the maths department, and then he taught in computer science in Auckland many years ago. Then he went to Liverpool for a while, and then he was at University of Queensland. He got a Federation Fellowship while he was at the University of Queensland. He's now at the Queensland University of Technology, QUT in Brisbane, and is a professor of computational mathematics there, but has had a strong link with Oxford, and particularly with Blanca Rodriguez, who many of us know at Oxford, and is going to talk about sources and effects of variability and heterogeneity in cardiac modelling. Kevin.
Okay, thanks very much, Peter. So I'd like to thank the ABI and Peter especially for allowing Pamela and I to stay here for a couple of months. We're on sabbatical leave. From here we go to Cambridge for 3 months. But we've been enjoying Auckland very much. We haven't been here since 1988, I think, more or less. Here permanently, here for a while anyway. So much of this work is done while I was, in fact, as Peter said, for 8 years I had 2 jobs. I was half-time at Oxford and half-time at UQ and QUT. So a lot of this work was done with the cardiac modelling group at Oxford, in particular Blanca Rodriguez and Maria Carusi, who's a philosopher, Vicente Grau, who's the husband of Blanca, and Nick Hale, David Kay, who are computational scientists, Alfonso Buenarovio, who's a postdoc of Blanca's, and some PhD students, and some clinicians at the John Ratcliffe Hospital in and also Szeged in Hungary, and also some clinicians at UCL. So, I like to get the acknowledgements done first because I usually forget to do them. So this is a short story that I like very much. It's by the Argentinian short story writer Borges. Most of you may know it, and if you don't know it, then have a read. It's a complete story. It's a complete short story. It's sort of nice. And then I can ask you at the end, why am I showing that slide? Okay, beautifully written. Right, move on. It's the most beautifully written slide there is in this talk. So. Okay, some issues in cardiac electrophysiology, coping with variability. And of course, variability manifests itself at many different levels and scales. I'm going to talk about 3 particular instances of variability. One is in ion channel dynamics, where we want to model the behavior of ion channels opening and closing, and ask the question, should we represent those in single cell models or not? So that's the source of stochasticity or variability. The second source of variability is at the tissue level, where we're looking at spatial heterogeneity. And probably all of us know that we can model the propagation of an action potential wave using the monodomain or bidomain equations. They're just reaction diffusion equations, essentially. The issue there is, how do you represent the spatial heterogeneity at many different scales and levels mathematically? And the third source of variability is at the population level, at the differences between us here in terms of our structures of our hearts, and how we account for that variability in our modelling. And what's the role of that in leading to things like personalised medicine? probably this slide is even from the ABI. I'm not sure, this picture. I'm not, so I've seen this in one of your, when I was up in your 6th floor the other day, I saw it in one of your pamphlets. So I don't know where it comes from, actually. But the point here is that we know that heart tissue is very heterogeneous. It's made up of all sorts of mixtures of all sorts of things, cells and fat and collagen and connective tissue and blood vessels in various proportions. Empty space. We model the propagation of electrical wave through that heterogeneous tissue. And we've used models, bi-domain models, monodomain models since 1975. And why? Because they work really well. But can we do it slightly differently? If we're going to do this in a personalised medicine setting, how should we use those models in a personalised medicine way? Should we be doing something else in terms of capturing the spatial heterogeneity? Here's some slices of tissue. You all know this. The second source of heterogeneity is between subjects. So this was a nice study done by Raf Borders, who was a PhD student of Blanca's Rodriguez in Oxford. And this is looking at the Purkinje fibres of rats. And here you see 4 healthy rats, the same age, the same sex. The Purkinje fibre networks are very, very different. Yet their hearts were more or less functioning in the same way. So, does it matter that our models don't necessarily capture that heterogeneity, that difference between the different rats? Or does it matter if we do or we don't? You might think it does if we want to go to some personalised medicine, if you change rats to humans, but maybe it doesn't matter that these are so different. This is a slide from Hugh Watkins' group in the BHF-funded Heart Centre at Radcliffe Hospital in Oxford. And it just shows normal myocardial tissue nicely aligned in sheets that wrap around the myocardium. And on the left, this is a study that they're doing on hypertrophic cardiomiopathy. So you see the disarray of the fibres. Of course, the heart enlarges, the fibres become disarrayed, and you have this heterogeneous structure. So if you wanted to understand that from a modelling perspective, what would your models look like at the tissue level? And this, you would all know, going into physiological variability from a slightly different perspective. These are measurements of ionic currents from the group of Barbara Cassidy. She's also at Oxford in the Radcliffe. They're very different at the human atrial myocyte level. Here's Purkinje fibres paced at a certain frequency. And you see the action potential has more or less the same shape, but the action potential duration can be very different from cell to cell. And at the whole heart, here's some ECGs of 2 patients, the same leads, and again, you can see the profiles are quite different from patient 1 to patient 2. Yeah, I believe that both of those patients were healthy patients. So there's all this physiological variability. Should you account for it? How do you account for it? And how do you account for it in your models? This you all know, so this is, I'm not going to go into this. This just talks about antiarrhythmia drug development. We all know that drugs have to be pulled before they go to market because they can induce different effects in different stratifications of population. So variability in biomarkers is a major challenge for risk stratification. And so how do we characterise that variability? What are the biomarkers that we use, and how do we represent it, and how do we then account for it in our mathematical models? And maybe it's important because it leads to these sorts of things. So, the hypotheses are that computational modelling offers a way to obtain sample-specific or patient-specific understanding of potential arrhythmogenic consequences of variability. That's just one small area of variability. Given that, then I would claim that we need new modelling approaches to capture this variability. Perhaps we need to do probabilistic simulation, that's sometimes called ensemble simulation, which they've been used in climate modelling for 20 years. Developing what I call populations of models, ensembles of models in the climate modelling area, with greater links to statistics in terms of how do you parameterize populations of models from experimental data. Then there are issues about what should our models look like? Should they be phenomenological? Should they be biophysical? Should they be both? And there's at the societal level, there needs to be a level of confidence for in silico models to be used in this sort of patient-specific area. I believe the reason that the French stopped doing atomic bomb testing in Maria, whichever one, was not because of any goodwill towards the people of the Pacific. It's just that their models had become so sophisticated and so well parameterized, they felt confident that they didn't need to do that atomic testing anymore. Maybe it's the same sort of thing in this setting. Okay, so these three levels of variability I want to talk about. So the first one is stochasticity in ion channel dynamics. So at the simplest level, a channel opens and closes. It opens, it closes, and ions flow in and out through this ion channel. In fact, what we know, there are various types of gates that occur in sort of modular form, and it's these collection of modules that work in complex ways for the channels to open and close. So there are very detailed, they're called Markov models, but they're actually state transition models that describe how these ion channels open and close, and fundamental work is done by many people. Johan Rudy is one. There's very complicated state models that describe calcium channels, sodium channels, potassium channels. So it's essentially a stochastic process because you have one ion channel here and another ion channel there. They don't behave in exactly the same way, and they open and close at different times. So it's essentially a stochastic process. So if you're looking at a small ensemble of ion channels embedded in the membrane of a cell, then the question is, what does the stochastic model look like that describes that ion channel dynamics? It's a different question if you then go to tissue level. So if you're then asking the question, how does the stochasticity in ion channels manifest itself at the tissue level when you see variability at the cell level, is a different question. Because diffusion, which is the fundamental role in ionic transport, smooths out the stochasticity in some way. So at the tissue level, maybe the stochasticity of the ion channel dynamics is less important, but at the cell level, it's certainly very important. And most experiments are done at the cell level, and you calibrate models at the cell level. So we did a small study where we looked at the IKS and the IKR channels. They play a very important role in the repolarization. And it's, from historical estimates, it seems that the number of ion channels of IKS and IKR type are relatively small compared with some of the other ion channel types. So we did a study where we took a cell model in its ordinary differential equation form, and we asked the question, how do we represent the stochasticity of the ion channels in that formulation? And without going into a long complicated story, because you have to write down the Markov model transition diagrams, and you have to build a stochastic model in the right sort of way, and there's some mathematics there. But at its fundamental level, it looks like this. So if I have a channel that's either in the opened or closed state, and I look at the open state, so Y represents the proportion of channels in the open state at any given time. And A and B are the transition rates, which are of course voltage dependent, so they're not constant. So these A's and B's depend on the voltage, and the voltage depends on some other things. But forgetting about that, then the stochastic model that you write down in that case, just for a channel opening and closing, has an additional term. So this is the standard ODE you would have. And then you have a 1 upon root n, where n is the number of channels. And you have this square root term here, which formally is positive. And dW is the stochastic equivalent of dt. W is a Wiener process. So a Wiener process is a stochastic process that essentially has Gaussian properties. Its expectation is 0, and the variance of a Wiener increment on some interval is the length of the interval. So that character, and Wiener increments that don't overlap in time are independent of one another. If they overlap, they're not independent. So that defines a Wiener increment, and that defines our stochastic model, ion channel model at the very simplest level. And then you need numerical techniques to simulate these things. And by the way, that's not so trivial because of this square root term. So you can imagine applying an Euler method, so the simplest ODE method, and extend it to a stochastic differential equation. What can happen is if you're near the boundaries of your stochastic model, and you're stepping through with an Euler method with a discretization of step size h, because this thing can be positive or negative with equal probability, you can step outside the boundaries. So then you have to ask the question, what should you do if you step outside the boundaries with your numerical scheme? So you have to do something. And the thing you have to do is you have to develop a technology around something called reflected stochastic differential equations. All that means is that you have to reflect back into your domain in the appropriate way. That's all. So there's a bit of nice computational mathematics there to do the simulations correctly. And press the right. I won't go into that. So the study we did was based around IKS and IKR. We did a block. We had some data in terms of both at the single level and at the tissue level. And we're looking at the role of EADs in this setting. So here you see, now where are we? Here's a control, there's no block at all. And here we do an IKR block. And now it's a stochastic model. And what you can see, the action potential has this variability that looks a bit like what we see in canine. What we tried to do is we had a single free parameter, and the free parameter was n, the number of ion channels of the certain channel types, and our model had a number of ion channels in it. But we focused only on IKS and IKR, so those were our parameters. And we tried to fit the data that we had against the experimental data at the single cell level. at the tissue level, you see this variability goes away. So at the tissue level, the stochastic fluctuations in the ion channel dynamics does not appear to be so important. But at the cell level, it is important. And so then we did a fluctuation analysis, and we tried to estimate the number of ion channels of IKS and IKR for both human and guinea pig, doing a fluctuation analysis. So this is there's no real way of doing this with a deterministic model, because the number of ion channels is sort of embedded in the model and you can't sort of get at it. But in the stochastic model, it's a key parameter in the stochastic formulation. So we can actually get a handle on estimates of the number of ion channels of IKS and IKR for human and guinea pig. Which, in a simulation sense, I think was the first time this had been done. Okay, so that was one study that we did at the ion channel level. The second study I want to talk about is at the tissue level. So most of us would know about the bidomain or monodomain equation that describes the propagation of a travelling wave in tissue. So forgetting that there, it's this here. This is the diffusion tensor. This is the voltage. This is the stimulus. This is the sum of ionic currents in and out of a cell, which are described by a certain type of cell model. It could be the Lua-Rudy 91 or whatever you want, the Grandi model 2012, whatever it might be. And so, augmented to this single reaction diffusion equation is a set of ODEs described by a set of ODEs described by these two equations, where the coupling is between this ion term here. And this ion is the sum of currents through the different ion channels, and where the additional terms are the terms that describe the behaviour of the ion channels opening and closing. So that's the monodomain formulation. You can have a bidomain formulation where you have intracellular and extracellular tissue, and you have an intracellular and extracellular voltage, and they're coupled in some way, so then you get a coupled reaction diffusion equation coupled for a system of ODEs. Then you ask the question, well, we looked at some pictures, the tissue is highly heterogeneous. Where does that heterogeneity manifest itself in these models? Where are they represented? Well, they sit in here, in the diffusion tensor. How do you evaluate that diffusion tensor? I mean, you need a diffusion tensor which varies in space, that accounts for that spatial heterogeneity at many different scales. Can you even measure those things? Well, let's assume you can measure them. So, if you can measure them, and I claim that you can't at very fine scales, but they go in there somehow. So, the greater that you look at the microscale structure, the more complicated that diffusion tensor becomes, and the harder it is to solve computationally, until you actually end up with the whole heart itself. So you replace the model by the heart, which is partly the point of that short story. So maybe there's different ways of thinking about it. So one way that I've been doing, mainly with Alfonso Buenarovio in Oxford and David Kay, is this is a reaction diffusion equation. It's a local problem. It's a local problem in the sense that if I take this outside, this is the Laplacian operator. Laplacian operator is a local operator. Essentially looks at neighbouring cells to evaluate it. All I've done is, well, assume this was constant, take it outside, replace the Laplacian by a fractional Laplacian. So this looks a bit weird, but now, so normally alpha would be 2. That would be a standard Laplacian. Now I have a parameter alpha, which is a number between 1 and 2. It's called a fractional Laplacian. And you might think that doesn't make much difference. But as soon as alpha is not equal to 2, you have what's called a non-local operator. So in other words, that means the influences there and there can influence what's happening here, both spatially and temporally. Okay, so, well, I've said all that. So I've said, use the cable equation, which is the reaction diffusion equation. You can write down the propagation of the voltage through tissue. It's coupled to a cell model, and you get an action potential that propagates. Okay. So what we've done is said, okay, this is a bit technical, I don't want to go into that. So in the very most simplest way is just to replace the Laplacian by this fractional Laplacian. So replace the value of alpha 2 by something between 1 and 2. And then ask the question, well, ask several questions. Why do that? Does it make any sense to do it? What does it mean biophysically? Or is it just a phenomenological model? And what is its effect? Can it offer additional insights into propagation of waves in spatially heterogeneous media, not just hearts? So, the biophysical justification. So when people talk about fractional diffusion, they often wave their arms around. They talk about anomalous diffusion. They talk about Levy flights and all sorts of things. So let me try and explain what. At one level, fractional diffusion is the following. So if I wrote down a reaction diffusion equation with a fractional Laplacian, that corresponds to the probability density function of a bunch of particles undergoing some walk process. And this walk process is called a Levy walk. And it's a jump process. So in actual fact, these particles are actually jumping. They jump, there's a waiting time, and there's a jump distance that you select from some distribution. And if you write down the probability density function of that ensemble of the particles, you get a fractional Laplacian problem. So then you might say, well, ions don't jump. So what is that a good biophysical justification for using a fractional Laplacian? So I look at it differently. I look at it in terms of potential theory, because that's what we're studying. So one can take a Laplacian, apply fractional power to it, apply it to an operator, and you can represent that in terms of some integral. So if n is 3, alpha is some number between 1 and 2. Okay, so this is a Riesz potential. And it works equally well whether it's a fractional Laplacian or a standard Laplacian. It's exactly the same. You can take the inverse of this thing. If you take the inverse of this thing, you get this integral here. So f has to have certain properties. It has to have compact support. It shouldn't be too broad. But nevertheless, you can write down an integral, but it's an integral over the whole space, over Rn. So it's a non-local operator. And our argument as to what this value of alpha is doing is that when alpha is 1, then this corresponds to a monopole in potential theory. And when alpha is equal to, that's just standard potential theory. And when alpha is equal to 2, that corresponds to a dipole. So either a 1 upon r or 1 upon r squared behaviour. And the values of alpha between 1 and 2 are intermediate between a monopole and a dipole. And why do they arise? they arise because the tissue heterogeneities establish a whole series of secondary sources at multiple scales. And you can add those up in some way. And if you wave your arms around, you can claim that this summing gives you a function of 1 over r to the 1 plus alpha, where alpha is between 1 and 2. So it sits between a monopole and a dipole. So that's our sort of justification of why fractional diffusion or non-local operators make sense. It's trying to account for this spatial heterogeneity in some way. Now there's no, if someone shows me a piece of tissue and I look at it and you ask the question, well, is alpha 1.93 or 1.76? I can't answer that question. I don't think some people will ask it of me, but I don't think it's a reasonable question and I can't give a reasonable answer. The reasonable answer is that there is no one alpha. In fact, there's a distribution of alphas. And our hypothesis later is that there is a distribution of alphas in different settings, in different stratifications of populations, in different diseases. Okay, you can do numerics on these things. I like to do that. So in actual fact, because we've got a Laplacian to a power, a fractional power, the eigenvectors are the same as the eigenvectors of the standard Laplacian, but the eigenvalues are the fractional powers of that. So you can do a spectral decomposition, and you can do spectral methods in this setting, and they work really quickly and they're really efficiently. And even though the operator is non-local, the spectral formulation works really well. So you can solve these things in a reasonable way. So then how do you calibrate? So we've just got 3 little studies of how we try and calibrate a monodomain model based on a fractional Laplacian. So the first is this work that comes out of the lab of Bardi and Bursak. I don't know them, but they're in the States. But what they did is it's a heart in a dish. They plated up neonatal rat cells in a dish, let them grow, and they can change the way that the cells grow into one another, the orientation of the cells and the structure of the tissue that ensues. So they can change that over a period of time, 3 weeks, I think. So they did that. And then having plated it, this was a random plating. So they stimulate at the center of the dish, and they record the action potential near the center and on the boundary of the dish. And what you see is this shortening of the action potential by some considerable amount. Now, if you do exactly the same thing with the monodomain equation, with I can't remember what cell model we used, but we used quite a recent one. You don't see this action potential shortening whatsoever. If you then take a fractional diffusion model, and you have to modify it in a certain way. We didn't get the units right, but when we do it properly, with a value of alpha equals 1.5, you see this action potential shortening. Now, what does that mean? Some people might say, well, you need to do something else in terms of your standard model, which is fine. Yes, you could do that. But we don't have to try very hard to see this action potential shortening when we use these fractional Laplacians. And again, we're not saying that there's anything magic about the value of 1.5. It's just with that value we see the shortening that's sort of consistent with the data. Then we took some data from Ben Hansen's group at UCL. It's about the relationship between action potential duration, the period of the travelling wave, and the activation time in human, dog, and rat. And you see this inverse relationship, which is thought to be important for healthy hearts. And again, what we did was we took a simple fractional model the fractional Laplacian, but we used the most up-to-date cell model we could find for, I think it was, I can't remember the models now, but they were very recent, the most appropriate cell models that we could find for human, dog and rat. And we did some simulations in a cable, just in 1D, that's all. So you can criticise this work, yeah, that's absolutely fine. And what you see is that with the standard models, which are the red lines, you don't see this action potential shortening again. You don't see this inverse relationship. But with values 1.75, 1.5, again you can see the shortening. There's some boundary effects here that you have to worry about a little. But nevertheless, these non-local models are demonstrating this inverse relationship that is known to exist. Whereas the standard models don't, unless you do something more sophisticated to them. And finally, we did a study with DT-MRI. This study was using some data from the Heart Centre up at the Radcliffe Hospital, where they were studying hypertrophic cardiomiopathy. You know that when you do DT-MRI, that the protons preferentially follow structures in the tissue. So the structures manifest themselves from the DT-MRI. There's something called an acquired signal, which is S of B. B is a parameter, which is called the B value, which is a mixture of things. D is the diffusion tensor that you're trying to estimate. And G is related to directions in which you apply the pulses. So this is all well understood at the DT-MRI. But we have a fractional model. We have a non-local model. So these assumptions are based on standard diffusion. So you have to modify this model in some way. You get something much more complicated, but where you have these fractional powers in the three spatial directions. So I call them beta 1, beta 2, beta 3 now. Okay, so you have to modify, you have to write down a different model to capture the anomalous behavior of protons following heterogeneous structures. So you have to do some maths. But this is what these three slides show what happens. So this is just in rat. These are just two slices. So the ADC is a measure that's called the apparent diffusion coefficient. So it just takes the trace divided by 3 of the eigenvalues of the diffusion tensor. So ADC, the sum of the eigenvalues of the diffusion tensor divided by 3. And you plot them at each point. And then there's something called fractional anisotropy. It's got nothing to do with the fractional that I'm talking about. But it's the square root of something that involves differences between the eigenvalues. And then we defined a new coefficient, which was the sum of these fractional powers divided by 3, similar to this. So now we have these 3 fractional powers between 1 and 2. We compute those at each point, and then we just plot those values. And without going into too much detail, we claim that this new AAC measure captures heterogeneity better than these other existing measures. And then what we did is apply this to 4 patients. No, 6. 3 normals, so without hypertrophic cardiomyopathy, and 3 with. And we did it in systole and diastole. So the ADC, remember, is just the trace of the diffusion tensor in terms of the eigenvalues. So you plot these at each slice spatially, and you plot the AAC, which are the sum of these fractional exponents, if it was a fractional model and a non-fractional model, all the exponents were, I've normalized it to be 1, everything would be red. So you see everything is sort of red here in this normal volunteer. In the diastole it's slightly different, but even so, it's basically red. But in the disease patient, in systole now, in systole before everything was red, now you see red and blue and bits of white. There's clear heterogeneity here. So remember, these are the sums of the fractional exponents, the powers, divided by 3. And this seems to be, so this part here seems to be particularly heterogeneous, and also in here. So this study is still in its infancy in some sense. But what we're hoping is that this fractional exponent is providing an insight into a spatial biomarker that doesn't really exist at the moment in cardiac modelling. So these fractional exponents capture the heterogeneity, and we hope that they say something about, in a crudish sort of way, something about that heterogeneity. And our hypothesis is that the metrics derived from the fractional diffusion can provide additional information in the diagnosis of structural abnormalities in cardiac tissue. And it's not that there won't be a single value, but there will be distributions of values in different settings. Am I going? Maybe I can rattle through. And the final little bit I want to talk about is populations and models. So this work comes from a paper that Yves Marder, who's a neuroscientist at NYU, wrote in 2011, talking about populations and models. So what's a population of models? So you take an ordinary differential equation, you have a set of parameters. Okay? So normally you have some data, you parameterize your set of ordinary differential equations against the data. Your ordinary differential equation is deterministic. We all know that our experimental data is highly variable. You measure something in one patient, it's this, and another patient, it's that. So how can you calibrate a deterministic model that way? Well, you take the mean of your data. So you take the mean of the data. You might not have many data points, but you take the mean anyway. You throw away all the variability, and you calibrate your model that way. Populations and models says, what I'm going to do is build an ensemble of deterministic models, calibrated against the data. So if I have some biomarkers, action potential, duration, APD 90, the height of the APD, height of the action potential, so some key biomarkers for sets of people, I have a range of values. Okay, so I have this range of values. So a model within the population of models, if when you run it with a certain choice of parameters, it's in the range of all the biomarkers that you have for the experimental data. Then you choose another point in parameter space, you run the model again. Does it have biomarkers in all those ranges or not? If it does, it's in the population. If it doesn't, it's not. So that's a population of models. It's a very simple idea. It's got nothing to do with stochasticity, but it's got an awful lot to do with variability. So I've sort of said all that, but the key thing is, the group in Oxford has written maybe 8 or 9 papers on using populations and models in different settings, and I've been part of 4 or 5 of those papers. The key question is, supposing you have 15 ion channel types, you've got 15 maximal conductances. Those are your key parameters. So you've got a parameter space of dimension 15. How do you sample that in some sensible way? You want to build an ensemble of models. Maybe there are several hundred of models in this population. How do you sample parameter space when your parameter space is 15-dimensional? So there's something called, this is where the statistics starts to combine, maybe a couple of minutes, the statistics starts to combine with the modelling and the numerics. So how to sample parameter space in some sensible way. So you can granularize, discretize parameter space. You know, think of a multi-dimensional Rubik's Cube where you just block space, parameter space up into different grid sizes, blocks. So there's something called Latin hypercube sampling, which I can explain in 2D, and I'm not going to go into this because it's more complicated. But here, there's two parameters, a range from A to B for parameter 1, and a range from C to D of parameter 2. I divide it up into 4 blocks. Latin hypercube sampling says place a single cross in each row and column. So I could do that like this. Okay, that would be a trial. A cross in that box means choose a point uniformly in parameter space in that little box. Okay, that's all that means. So that would be a trial. Now you could repeat that trial again and you'd get a different trial. So you could do multiple trials, or you could have a much finer granularity here. So have a much smaller granularity. So then you choose a point in parameter space, run the model. Does it fit all the biomarkers in the range? Yes or no? You might look at it and say, well, I'm not very happy with that because it hasn't. What about this space over here? I haven't even looked at that space. So there's something called orthogonal sampling, which would guarantee that you get one cross in this 2 by 2, this 2 by 2, this 2 by 2, and this 2 by 2. So it's a sort of variant. And so then you have lots of arguments with people who say, well, I don't like this, and lots of arguments at Oxford with different people say, I don't like populations and models because how do you guarantee that you sample parameter space adequately? And my answer to that is, well, you're using a population of models with one model in it. I think what I'm doing is better than one. That's the flippant answer. The non-flippant answer is to try and do two things. One is try and prove theorems about coverage, which Pamela and I and some other colleagues at UQ have done. Well, finally, is to become a card-carrying Bayesian statistician and try and do something clever with building populations and models where you can actually think of balls in parameter space that you move around using some sort of MC approach that coalesce on some distribution that represents distributions where parameters are likely to lie that fit the biomarker ranges. And we've just written a paper that does some Bayesian MC approaches in that setting. Okay, so putting it together and then I finished. That one is, it's sort of obvious, I'm not going to go into it. It's the relationship between what are the questions, what are the data, what sort of populations and models do I want to build, and how do I calibrate them? If I only have small data sets, I can calibrate against a range. If I have lots of data, maybe I can calibrate not against the range, but calibrate against the distribution. In other words, if I have 500 people in my test, I've measured this biomarker, I see a distribution of values of the action potential, then I try and calibrate my population of models against that distribution. that I have in the data. And what that actually does is to remove some of the floppiness in the model, in the populations and models. Okay, so last slide. Oh my God, it's very dark. Okay, just some final thoughts. So, no, you know, these aren't. We all think about these things. What do we want from a model? But the second question: should we add increasing complexity just because we can? But in some cases, you should. But some cases, maybe you should look back and ask, well, what are the fundamentals that underlie the mathematics when we build that model? Does it really apply in this spatially heterogeneous setting or not, or should we do something else? On the other hand, I've probably not described particularly well what this fractional Laplacian is really doing. It's not intuitive, so should a model be intuitive? That's related to how do we trust a model? If it's not intuitive, how do you go about trusting it? And then if you can't trust the model, what does that mean for doing patient-specific modelling? And finally, what I have learned is that when I started doing this work, I knew very little statistics. I never liked statistics when I got taught it here at Auckland for whatever reason. So I just pretended it wasn't there. But I found that more and more now I need to understand, because I'm doing probabilistic simulations, I need to understand more about Bayesian statistics. And I strongly believe that in our modelling and simulation work there needs to be greater links to the modelling, the simulation and deep statistical analysis. Not just in analysing the data, but in building these populations of models. Okay, that's it. Thank you.
Okay, maybe I could start with a couple of questions that occur to me. Kevin, one is, when you show the way that a fractional derivative for your Laplacian or your fractional Laplacian can give you a better result, for example, the shortening of an action potential, etc., is it the same value? Can you do experiments on the same bit of tissue where these different experiments can be shown to be better represented by the same value of alpha? Because I saw your fractional alpha.
Yeah, so
yeah,
no, I understand. So we haven't done that, but I think that's. I mean, it's a good suggestion, but in some sense it's a bad suggestion, because what I really believe, it's not a value of alpha, it's a distribution of alphas. So the question really is there some distribution of alpha that's not just between 1 and 2 uniformly that can better describe a set of experimental...
But is your distribution of alpha just reflecting different heterogeneity, characteristics of heterogeneity in different regions of tissue? If you just take one small region and characterize it...
Oh, I see what you're saying.
Then you come up with a particular value of alpha.
Yeah, so you can homogenize over a particular value of alpha and come up with an alpha which is homogenized over the spatial region. We haven't done that yet because we don't know how to do it mathematically. But we can just take averages, for example. So you can do that. But if you do that, you also lose the value of alpha as a probe to probe spatial heterogeneity in certain subdomains, which is why I think it was important in this hypertrophic study, because we were really seeing it's very hard if you take a standard model to capture that spatial heterogeneity in certain domains of the heart. But with this fractional exponent, you seem to get greater insight. So if you'd homogenize it, we'd sort of lose that insight.
Question, Marty.
So thanks, that was interesting. Just on that last point you made, I mean, I was intrigued by your comparison between the ADC and the fractional anisotropy and the fractional diffusivity, I guess. diffusion.
Yeah.
Which one do we believe, though? I mean, is there independent histological evidence that suggests that indeed that heterogeneous distribution is something we really should be looking for? Do you see what I mean?
Yeah, no, I mean, I don't know, so this is a deep question, what do we believe? I'm not sure I can answer that one in a few minutes. I understand I mean, sometimes you demonstrate things by the contrapositive. So, I mean, it seems to be that without a great deal of effort, we can capture things with these fractional Laplacians that standard models cannot capture. Now, that doesn't mean to say that standard models cannot capture it, but you may have to do something else. But I don't know what that something else is. I mean, but I'd be quite happy to try things. So that's suggestive, at least. The second thing is that these fractional exponents have the potential to provide spatial biomarkers that standard models, again, cannot provide apart from the diffusion tensor. And of course, that's very hard to measure. So again, there is a potential there for this to be a biomarker, but this is very early days. So who can say? I mean, can we get enough patients, hypertrophic patients, to do a very detailed study and say that this is a meaningful biomarker in this case? I don't know. It's a good question, so I can't answer it. But these things, at least to my mind, they're suggestive.
It's really nice to see how the fractional Laplacian is able to reproduce some of the behaviours that you see. But I'm worried that a fractional Laplacian operator is used, but at the expense of locality. The physicist in me leaves me scratching my head.
Yes, I know.
Should I accept that?
Yes, no, and I spent many weeks and months thinking about what does it mean biophysically. And one way to think about it is that associated with these fractional operators are heavy tailed distributions. So heavy, so if the relationship between the exponential function and pure diffusion, there's the exponential function here. So in the fractional case, you don't have this, but you have a heavy tail. So what does that mean? It means that an ensemble of particles are more likely to be further out somewhere than in the standard diffusion case. How might that happen? Well, I mean, ions don't hop around as far as I know. But what they can do is they can take different complicated connected pathways through the tissue. So the spatial heterogeneities actually form different sorts of pathways through the tissue. That's what the heterogeneity does. So some ions can take pathways in some directions and some ions take pathways in other directions. So it's that spatial connectivity in very complicated ways that can lead to these heavy tail distributions. That's my hypothesis. So it's not that they're not jumping, but objects are taking advantage of spatial structures in complicated ways.
So another way of perhaps looking at that is it's somewhere between a homogeneous, maybe 3D distribution and something that might have lower dimensional structures,
yes.
1D and 2D.
Indeed. I mean, that's why when I spoke about monopoles and dipoles, it was in that flavour. It's a similar idea. Yeah, no, I absolutely agree. I struggled. I had lots of discussions with people about these things, and I don't want these to be phenomenological. They should be biophysical. Therefore,
that's the question that gets raised.
Absolutely, all the time. Well,
that wouldn't worry about it.
Well, they should do. Well, I certainly worry a lot about it. But the justification, the discussion we just had there is sort of close to the mark. And it was when I was talking about this potential theory, it was in that same spirit. So it's the structure itself that creates connecting pathways. That means that if you actually look down on it from above, you would see heavy-tailed distributions.
From a computational point of view, you're going from a diagonally dominant matrix to a more general...
To a full dense matrix, where the weights tail off.
So what do you find in practice is the relative expense of solving those?
Okay, so let's see now. They're dense, so you've got to do dense linear algebra. So if you're in regular domains, the spectral methods work brilliantly. But they sort of need to be regular domains, so you know something about the eigenvalue structure. If the domains are irregular, then you have to do some clever linear algebra. But it turns out we've developed some preconditioners that work really well. And then because you're dealing with long vectors, it vectorizes beautifully. So in actual fact, you can solve them really quite efficiently if you do the right sorts of things.
Actually, following up the same problem. So I was thinking that in the derivation of your Laplace, you actually assume sigma to be constant. And then you replace your Laplace as a fractional Laplace. Won't you get the same effect if your sigma has been a function, but that function also have a not a polynomial but a polynomial, and then you get the divergence of that polynomial functions and you get the derivative.
Yes, I understand. I mean, it's a very good question. There are theorems that say that in some settings, no matter how, what you're really saying is why not have a nonlinear spatial tensor? And there are theorems that say that in some settings there is no nonlinear spatial diffusion tensor that you can write down that will give you the same properties as a fractional problem. And there is such a theorem. That you might say, well, you know, do those examples arrive in practice or not? That's a different question. But there's not necessarily an equivalence between nonlinear diffusion theory that you're talking about and the fractional approach. It's a good question. But a second question is, even if you could do it, how do you write down this non-linearly spatially varying tensor from measurements?
Yes, but then the argument is that this is more intuitive.
Well, I'm not sure it is more intuitive. It depends on how much you think about what a non-local problem is. But they're not equivalent. They're not equivalent. There are theorems that say that you can't do what you're suggesting.
Right. But then, if you have come up with a nonlinear, say you do not know why or how, but you manage to come up with that,
yes,
you can retain the local interpretation. Then we do not need to go to non-local where the intuition and the interpretation is fuzzy.
See, okay, so you're not the first person to raise these questions. So the first question is, you could do that, but first of all you have to calibrate at many different scales your nonlinear diffusion tensor. So that's already a hard problem. So how do you do that against the data? Secondly, you're right. These fractional models are not intuitive unless you think about them deeply, but they're simple. I don't need a complicated diffusion tensor to replicate the sorts of things that you would do via a very complicated nonlinear diffusion tensor. I don't need to do that. So, yes.
Okay.
Good question, though.
5, maybe we should stop, but join with me in thanking Kevin again. Don't forget that there's food and drink.